Thank you, everybody. And first of all, thanks to the organizers for this wonderful workshop, and thanks to all the speakers. I learned a lot. I'm mainly theoretical physicist. I am working on biological systems, and this game theory and evolutionary game theory somehow I enjoyed a lot, and somehow it is connected to some of my previous works when I was working in my home country. Today I will talk about global epistasis for a very simple systems like elastic network. And this work is in collaboration with Josh and Andrea Liu, and Felipe Martins is a PhD student at the UPenn in the physics department. So let's start. For a protein, we know that the sequence of amino acids, it gives us the structure of that protein, and the structure somehow dictates the function. For what's the meaning of term of epistasis? For example, consider this wild type protein, and if we make some mutations on it, for example, we change this amino acid and measure its function. So its function could be changed like by delta F1, and at the next time we perform another mutation on another amino acids, and it changes its function by delta F2. If we, at the same time, we perform both mutations together and measure its function. If the effect of the mutations is non-additive, people call this phenomena as epistasis. So we can define some metrics showing the different mutations, and the components of the matrix, if becomes 0, it means that the effect of mutation is simply additive, and if they are not 0, it means that we have some kinds of epistasis. In experimental point of view, people can use different methods in order to calculate or to measure the epistasis metrics, like using a deep mutation scanning. I don't go to details of this kind of experiments, and if you are interested in some details of details of my talk today, we can discuss later. And so, for example, this is a real experimental data. It has like 500,000 double mutations, and they measure this kind of epistasis metrics. Assume that our system, our protein, behaves has an epistasis behavior. If there is no connections between different epistasis, in that case, we don't know a lot the effect of different mutations in the system. And if we look at the fitness landscape, it would be a little bit very rugged. But somehow, if there would be some function relate different mutations to each other, we would expect to see a little bit simpler fitness landscape. And people call this kind of epistasis global epistasis. I will explain why we biologists use global in the next slide. For global epistasis, Josh and his group made a very beautiful model to analyze this kind of epistasis. They say that, okay, if we have the position of the amino acids and the amino acid in position I, if we show it by AI, and after mutations, we can consider the effect of all mutations in terms of some additive terms like beta IAI, which shows that if amino acid AA seated in the position I, in the global epistasis behavior, there will be a non-linear function connected the underlying linear behavior in our system. So the fitness, or for these days we can use for function of a protein, this nonlinear function of the linear trait, somehow it is connected to the function of that protein. So because of the presence of this nonlinear global function, people call it global epistasis. To understand the origin of this global epistasis, we use a very simple model for this kind of real biology problem. For this map, consider for a real protein on the left side and our simple model in the right side. The sequence of amino acids gives the structure and it gives us the function. The function of the protein should depend on the location of the amino acids and the sequence of the amino acids. For us, we consider it a little bit more simpler. The sequence of the spring constants for a given network, it gives us the function of that network. I will explain what I mean, the function of the network. And to make a similarity to the real protein, we use the 20 distinct spring constant for this network. Okay, for this model is a very good model for some kinds of protein, like protein allostery. And for the protein allostery, we have a regulatory part or source part, and the response is far from that source part, and we call it a target part. So it's a kind of input-output problem. The input could be considered as a local strain in the input part, and the response of the protein can be considered as a strain in the output part. So we have a kind of input-output problem. So for our network, we initialize a network with a given spring constants and a given rest length for each spring. And what is the task, or what is the function of this network, similar to a protein? We apply input strain in an input edge like this, and measure the output strain for far from that input, like here. And if our network trained to give us a specific desired output strain, we will call this network is trained for that specific function. So it is similar to a wild type protein. Okay? How we can train it, I don't go to the details, but it is a kind of double optimization problem, because at the same time, we should minimize the elastic total energy of the system in order to fulfill the force balance and torque balance. At the same time, we would like to minimize this loss function. The loss function is just the changing of the measured output strain and the desired output strain. So if we train our network for a specific function, we will get the loss equals 0. So at the same time, our energy for that configuration is minimized, and I mean the total energy of the elastic network. And the function of the network can be considered as any function, mathematical function of this loss quantity in our network. We use this simple function, apply a very simple value plus the loss function, and take a logarithm of that. So it means that we can train our network, and we can get the wild type network like a wild type protein. So what is the advantage of this? We could tune the mechanical network, and one of the questions could be that, okay, which edges is the most important in that network? In terms of the protein, which mutations is the most important? And can we predict the effect of mutations in our system or not? To do that, we carry out two-edge, one-edge, two-edge, higher order mutations in our system, similar to the deep mutation scanning, and measure the output function. And so we have a genotype-phenotype map. The genotype will be the set of spring constants corresponding to each edge. And the phenotype will be the function of that network, which depends on the strain on the output edge. Similar to a real protein, we can apply global epistasis analysis. So we will have additive effect for edge I if the spring constant is Kj. And we can find this inferred additive trait, let's say. It's a function of all the additive terms. And ask ourselves, ask the system, what is that nonlinear function of those inferred additive trait? In order to do that, we use Maven package. One of the package is suitable for the global epistasis. But just to mention that for a small network or for a small protein, we will have like 60 amino acids. We will have 1,200 free parameters for this part, and some nonlinear functions here. And you can consider it is like 20, 30 free parameters for that part. So, sorry, for this simple problem, we have like 1,300 free parameters. So in this package, we use some kind of gradient descent algorithm for machine learning, and it takes time and RAM you can imagine. So what is the result of this? For example, for one example, this is the outcome of that global epistasis analysis. And here the line shows the edge index, and here I showed the spring constants similar to different amino acids. And you see that in some edges, the effect of mutations are very important. And the gray circles shows the wild type amino acid or wild type spring constant. So it means that the edges with more red color, they are more responsible in the changing of the function of the protein for that specific task. So it is very important. We could make a simple model similar to a real protein. And we see that simple model gives us a behavior like a global epistasis behavior. And we could find that nonlinear function by applying that machine learning method to find that nonlinear function. So what we learned that the elastic network behaves like a global epistasis. That's great because we know something more from physics about the elastic network. If we train the network around and we can expand the function around its minimum. The minimum of the function L is 0. So the first leading correcting term after perturbation comes in this second order of changes, second order of perturbation. And we see the very beautiful matrix here. We call it loss Hessian. So we can find the loss Hessian from the network analysis. And for the loss Hessian, we can calculate its eigenvalues spectrum. And if we get a small eigenvalue, it means that if I go to the direction of the corresponding eigenvectors, I don't see a large curvature in that direction. But if I get a very large eigenvalue, it means that it is corresponding to the very high curvature direction. What does it mean? It means that if I perturb our system, I mean in terms of the mutation, along that large eigenvalues direction, small changes make us large changes in the function. So it is good. We can use this idea in order to predict the key edges in terms of the mutation. Okay, we use this idea for that shown specific network, and this is the result. I applied for a very large strain, for example, in the input side, and the desired output is a large strain in this output side. And this is the spectrum of the eigenvalues. There are a few eigenvalues here, like 10 to the power minus 12. So it means that, but we have only very large eigenvalue correspond to this spectrum. So it means that we can estimate the behavior of the network based on this very large eigenvalue. And it implies another things that we are in the over-parameterized regime. I will go back to this part a little bit later. So we use the same idea for different networks. I showed here only for 20 independent network, and I applied for changing the structure of the network and changing the initial conditions. And I have those data as well. So we see that there is a very good agreement between the Hessian analysis based on the purely mathematical physics and the global epistasis analysis using some kind of machine learning technique is a statistical analysis. And the key edges are light here, and more or less we could capture the key edges using the Hessian analysis. This is the Spearman rank correlation, and you see it is like the maximum value is like 0.8, but for the majority of the edges, the value of the correlation is larger than 0.7. And we can analyze for the overlap fraction, and this is the overlap fraction of the cost Hessian and the global epistasis for different networks for different tasks. And the value is very great, and it means that there's a good agreement with the cost Hessian analysis and global epistasis analysis. But the next question could be that, okay, we see that correlation. Can we somehow predict the key edges or key mutations based on the structure of the network or not? To answer to this question, we analyze the susceptibility. How it comes? Somehow the loss Hessian is defined as the changing of the loss in terms of the changing of the spring constant. What about the physical Hessian? The physical Hessian can be defined as a changing of the energy of the system when we a little bit perturb the location of each nodes. And it is shown in this beautiful paper that the loss Hessian can be related to the physical Hessian by this a little bit big equation. And what are the quantities are Si alpha? Actually, it depends on the physical Hessian, and it depends on the incidence metrics. So that part of the relation purely comes from the architecture of the network. And this part comes from the input, output edge, training data, and which depends on the function. So it means that if we make a connection, we see that there's a connection between the structure of the network and the loss Hessian. And so maybe we can use the analysis of this part and compare to the analysis of the loss Hessian comes from that part. This is the result of that comparison. Here we compare the edge susceptibility I showed in the last slide and the stiff mode of the loss Hessian. And we see that there is a good correlation, at least for the first 20 or 30% of the edges. So here we compare the importance of the susceptibility and the global epistasis. And we see that there is a very good correlation between them as well. So what does it mean? It means that we can capture key edges from the high susceptibility edges. The main result of this work can be summarized like this. And I only emphasize on the last line. Maybe using the similar method, and we make more predictions. And in that sense, there is no need or less need to the deep mutation scans, which cost a lot from the experimental data. And at the end, I would like to thank Felipe, Josh, and Andrea and the finding. And thank you very much for your attention.
Thanks, Farshid. Any questions? Yeah.
Oh, very simple question. What does the node represent in your Spring network?
In our Spring network, it doesn't represent anything, but for a mapping to a real protein, each node can be responsible for each amino acids, and the edges could be interactions between two neighboring amino acids.
But responsible doesn't mean that the node does represent the amino acid.
Yeah,
no.
Yeah.